Welcome to lesson 1 for module 9. This lesson specifically is going to go over cell size and chromosomes. Module 9 is all about cell division and reproduction. So learning objective for this one is that you'll be able to explain the importance of cell size and chromosomes. So just 3 new vocab words for this lesson specifically, and that is chromatin, chromosome, and nucleosome. So let's talk about cell size limitation. So what we're looking at here is ratio of surface area to volume. So surface area being the outside, volume being the inside. So several factors influence cell size. Surface area is the area covered by the plasma membrane, what you could touch theoretically on the outside. And then the plasma membrane is the structure through which all the nutrients and waste products must pass. So we did a whole lesson just on the plasma membrane. It's the semi-permeable membrane. Some things can go through on their own. Some things need to be transported. But everything that needs to get in the cell or out of the cell has to go through that membrane. And then volume is the space taken up by the inner contents of the cell. It is the space that it takes. So the ratio of surface area to volume decreases as the cell gets bigger. So we have here a ton of surface area to a smaller amount of volume. Here, both the surface area and the volume get bigger, but if we were to do the geometry and calculate volume here versus the surface area, the ratio goes down. So here we have less surface area available to more volume, and then even more here. Surface area is still bigger, but when we compare them, the ratio here, we have a lot more volume than we do compared to the surface area. So cells with a higher ratio of surface area to volume can sustain themselves more easily. If we have waste even in the middle of the cell, it can really easily get out. If there's waste in the middle of the cell, it's going to take a lot of energy and time to get that out of there. We also have to think about transporting substances. So smaller cells can transport substances more easily to any side of the cell. It's not always going to want to get to the closest end. Sometimes it needs to transfer across the cell over to a neighboring cell. Diffusion is inefficient over longer distances. So diffusion works really well if it's just like from here to right over there. But if we have to go cell to cell to cell, then that's not the most efficient. The cytoskeleton transportation network, it's like these little spindle fibers that move everything, becomes less efficient for a cell if the distance to travel becomes too large. They have the energy to move things maybe longer distances more efficiently than diffusion, but they can't go very, very far distances. And then cellular communication, it is also more efficient in smaller cells. Chromatin and chromosomes, last thing we're going to go over for this quick lesson. So chromatin is the relaxed form of DNA. When we think about DNA, it is those double helixes. We will learn more specifically about DNA, but DNA is a double helix. Chromatin is a relaxed form. Chromosomes are condensed structures that contain the DNA that are visible during mitosis. So we're going to learn all about the stages of mitosis, and the things that we see separating during mitosis are going to be those chromosomes. Chromosomes are passed from generation to generation, and eukaryotic DNA is organized into chromosomes. So our DNA organized into chromosomes. Prokaryotes, not necessarily. So the phosphate groups in DNA, because DNA has a phosphate group, create a negative charge which attracts the DNA to the positively charged histone proteins to form a nucleosome. It's a little bundle of DNA. The nucleosome group together into chromatin fibers, which supercoil to make up chromosomes. I have a graphic to show you on the next slide right here. So this is DNA. This is what we think of right here. This is true, a double helix DNA. They make little, or have, they coil up around these little histones, and that becomes a nucleosome right there. And then as those coil up more and more, we get a chromatin fiber. And then as those coil up even more and more and more and more and more, we get a chromosome. So in the chromosome here is the centromere and the chromatids up there. But that is super, 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 super coiled DNA. And that is the end of this lesson. So next lesson, we'll go over mitosis stages specifically. If you have any questions here, please let me know. I'm not expecting you to know a whole lot about the structure of DNA quite yet. We will go over that specifically in another lesson. But you do need to know DNA coils up around histones to create nucleosomes and eventually coils into our chromosomes. Let me know if you have any questions.